day. And for next 5 days, like we will be talking about all the integrities of stem cell technologies that is happening right now. And you will be learning about stem cell banking, stem cells, hematopoietic stem cell transplantation, adult mesenchymal stem cells, what happens in an industry of stem cell banking, what happens in stem cell research, what we do in lab, how different are we from other people, and all that. So for the first slide itself, like it has a clear message for everyone. Be like stem cell, differentiate yourself from others. So this is going to be a session for next 5 days, wherein like I want you all to start learning rather than studying. So what's the difference between learning and studying? Hello. So can anyone tell me the difference between learning and studying?
So learning is when we read, whereas studying is when we cultivate it by ourselves.
Okay. Yes, that's somewhat right, but still, like I'll tell you, like you all must be knowing how to ride a bicycle, right? So if I tell you, like from today for next 30 years, like you should not be riding a bicycle, and then only exactly after 30 years I give you a bicycle, you would still be able to ride it because you have learned how to ride a bicycle. Okay. Similarly, when I tell you like what you learned 10 days before, what you study 10 days before, you'll not be able to recognize it or even keep it in memory. Or even if it is there, it must have been in a peripheral memory and then it must have washed out. So the first thing that what we are going to do from today till Friday is our concentration is to learn things and study through learning. So that is going to be the difference. And by doing this, you are going to differentiate like stem cells, and you are going to be differentiating yourself from others in the process. So with the first slide, with this message, I'll be taking further steps to get into the process. Okay. So this is basically like you all must be knowing and heard of stem cells. Like you must have heard about stem cells. Like they do a lot of work in the body and they differentiate and all that. But to make it very simple for you, like there are two slides which basically talks about what is stem cells and what is stemness. So there is a difference between a stem cell and a stemness. So stem cell means it has to differentiate, as you must be knowing it, and it has to also self renew. Okay. So in that case, so if you see in the first slide, like you have one cell and then like it divides into two. So what is this process of dividing equally into two? That you must be knowing. It is like mitosis, and the cell divides and then it gives rise to two cells, two daughter cells. Okay, so stem cell has to have a capacity to self-renew in a way that it has to keep uh, doing a symmetric cell division. But at the same time, like it also has something to do, not just self-renew, but it also has to differentiate. So in that way, like if you see this slide, like you have one cell and then it differentiates and then it becomes two different cells completely. So the property of stem cell is to first self-renew and differentiate. So what is stemness means? The stemness is a capacity of a stem cell to differentiate as well as to self-renew. So this concept has to be very clear in your mind.
406572.
Yeah. So this concept has to be very clear in your mind before we get into the bigger concept in stem cells, what we do, what is cell grafting, what is cell engineering, what is cellular immunology, and all that. Before we get into the transplant and types of stem cells and everything, you need to make sure that you know this difference between what is a stem cell and what is a difference between stemness and stem cell. So with this clear picture that the stem cells will renew as well as differentiate, we are going further. So what happens, this is same pictorial representation. You can see like it is self-renewing, so that like we don't run out of stem cells. So why do we need stem cells? First, the body has to make different kind of stem cells, and different kind of cells. It has to make heart cells, liver cells, kidney cells, epithelial cells, endothelial cells, all germ layer cells and everything. So for all that, you need something called stem cells. That is the reason why the stem cell has to self renew, and then same time it has to differentiate. So why it has to differentiate? That is to replace the dead or damaged cells throughout our life. So like there was this process helps somebody like somebody like us to make sure that while we are living our life, the internally the stem cells are doing the work of self renewal as well as differentiation. Now, like all stem cells, they do not differentiate into every type. So it is not something like one cell is differentiating into liver cell also, cancer cell, like kidney cells also, heart cells also, as well as like skin cells also. It is not like that. So there is something, a process which happens so that the cells are differentiating into, not every stem cell is differentiating into every type, but in a specific type. So we will be learning that now. So what happens here is like you have a stem cell, it has differentiated into a skin progenitor cell, and then from there you get a skin cell. Okay. And similarly, you have a stem cell which has a neural progenitor cell, and then you have a neural progenitor cell, and then it differentiates into a neural brain cell. Okay, neuron, which is a brain cell. Now, like the earlier slide, we saw that it self renew as well as self differentiate. Okay, but now it does not differentiate into every type. Why it does not differentiate into every type is there are reasons for it, which we'll be learning in further slides. But before that, this is an example wherein like the skin stem cell first gets differentiated into a skin progenitor cell, and that from that skin progenitor cell it gets differentiated into a skin cell. Now what is progenitor cell? The progenitor cell is a cell that can differentiate into a single lineage or a single type. So this, if we are talking about skin progenitor cell, it can only give rise to skin cells. It can't give rise to a neuron or a brain cells. Similar way, when we have a neural progenitor cell, it can only give rise to neurons and not skin cells. So this is something called progenitor cell, and we would be learning more about progenitor cells in next 5 days. And you have to have this concept very clear that every cell of stem cell doesn't differentiate into every type, and there are ways in which the stem cells will be differentiating. The first is a progenitor cell that can differentiate into only one type. Okay. Now, so this, the first cell that we are talking about over here on the left is like on the left over here is basically an embryonic cell, and from this cells it differentiating into a skin progenitor cell and a stem cell. So whenever there is a differentiation happening, one cell will remain a stem cell and other cell will only differentiate into a different cell. In that way, like you have the stemness maintained in a way, it is for self renewal as well as differentiation. Now, similar way, like we have now learned how the stem cells are differentiating into a progenitor cell. From progenitor cell, how we are getting a specific cell type. So this diagram will give a better picture of the same explanation, wherein like we are going one step further of learning what is multipotent progenitor cell and what is unipotent progenitor cell. Now in this cartoon, you can see that you have a stem cell, which is self-renewal capacity it has, and it has become a multipotent progenitor cell. Which multipotent progenitor cell means it can give rise to different types of cells, like different types of same lean cells. In that way, like it is here, over here you can see, like you have this cell as well as this cell, both are same, and it both are differentiating into a same lineage. Wherein here in case of progenitor cell, like it is specific only for one cell, it is dividing into only one cell. Wherein in case of multipotent progenitor cell, it is dividing into different cell types. Like one you have a dendritic cell, one you can have a monocytes, or one you can have a neutrophil or a monophil or a monocytes or different types. So in that way, like what happens is a multipotent progenitor cell will give rise to different types of cells which are coming from one multipotent progenitor cells. But in case of unipotent progenitor cell, it is dividing into only one specific cell. So this is basically the difference between a unipotent progenitor cell and a multipotent progenitor cell. The multipotent progenitor cell is giving rise to different mature cells, where in case of unipotent, it is giving rise to only one specific mature cell. So now with this concept, we further get into the type of stem cells. So you must have heard about pluripotency, totipotency, multipotency, and then like induced pluripotency and all that. So to make it very easy for you to remember as well as learn, like we have made it in a way like this particular slide will give you an easy example of the differences in different types of stem cells. So the first is the embryonic stem cell, which is basically a pluripotent stem cell, which can form almost any cell type in the human body. Okay. And in case of tissue specific, like I said, like brain or like liver or skin, they are all called adult tissue specific multipotent stem cells, can only form a limited types of cells. In case of here, you can see like this multipotent stem cells is giving rise to blood. If we are talking about blood cells, then it is giving rise to, like imagine this is neutrophil, this is monocyte, or this is dendritic cells. But it is not giving rise to a new cell, which is a skin cell, because this is limited to a particular lineage. We will study what is lineage further. But with this concept of multipotent, it can only divide into a limited types of cells. If you're talking about blood, it can divide into neutrophils, like granulocytes, monocytes, B cells, T cells, and everything. So how they are dividing, we will be learning little further. And induced pluripotent stem cells, you must have heard this, induced pluripotent stem cells. So who is Yamanaka? Do somebody know like who is Yamanaka and what is Yamanaka factor? Hello? 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 Are you guys there?
Sorry, sir.
Yeah. What is, who is Yamanaka? Because everything that you are going to do in future and everything that is going to happen in future, wherein like like every cell is getting differentiated into every cell type, and we are going to go beyond the horizon of the limit that we have set as of now in regeneration medicine. It's all going to be broken. It is already getting broken, but it is all going to be a different ball game altogether, only because of a man called Yamanaka, who actually made a process wherein like he studied that when he is changing an embryonic stem cells by inducing 4 factors of a cell, that can differentiate into any type of body cells. So embryonic stem cell, they say that, like I said, embryonic stem cell means they can form almost any cell type in the body, and that is pluripotent. So this is a natural phenomenon that is happening inside our body. But induced pluripotent stem cell is something, he took that embryonic stem cell and he induced it to become a pluripotent stem cell, which can differentiate into any type of body cell. So why did he do that, and what is the relevance? By actually making this cells differentiate into any type of body cells, you are making sure that you are getting more number of cells and more number of specific cell types that can do the work, rather than waiting for a cell to divide into a specific lineage. So what is this Yamanaka factor? These are the four factors, like these are the four factors which are all proteins, and they are like very important for the embryonic stem cells. And these factors, when they are expressed or induced to express more, then the embryonic stem cell will differentiate into an induced pluripotent stem cell, which then can give rise to different types of cells in our body. Now, like, we have learned what is pluripotent, which means like it can form almost any cell type in the human body, and multipotent, which can give rise to a limited type of cells in the body. Like if you are talking about skin, it will give rise to all skin cells like fibroblasts, keratinocytes, epithelial cells, and endothelial cells, skin endothelial cells. And if you're talking about blood, then it can give rise to monocytes, lymphocytes, B cell, myeloid lineage, and then like lymphoid lineage and stuff. And in case of induced pluripotent stem cells, it can differentiate into any type of cells, and they are induced by scientists in the laboratory so that it can differentiate into any type of cells. Now, what is totipotent? Totipotent is little different from pluripotent, wherein in case of pluripotent, I have written something called almost. Okay, but in case of totipotent, it can differentiate, one cell can differentiate into an entire organism. So that is a difference between a pluripotent and a totipotent. In case of totipotent, it is like one single cell can form into a complete organism, like how we were born, like just a cell, and then like we became a embryo, and then we got differentiated into a fetus, and from fetus we came into the earth, and then became a full human being, a full adult. So that is something called totipotent. Now, so the same concept we are getting further to make it further easy for you to understand. Like what happens is in vitro development, how it happens. Like we are talking about cells getting differentiated, cells getting renewed, and then like cells getting differentiated into specific types and everything. So in that way, to make it little easier for you to learn the process, what happens is, imagine like a sperm is getting injected into an egg on day 1, and after 12 hours, you can see on the right, it has formed 2 cells. Okay, and in day 2, it has become a 2-cell embryo. And then in day 2, after few hours, this 2 cell has divided into a 4 cell. Okay, and on day 3, it has become an 8-cell embryo. And then in day 4, it has become a morula. Like all these cells have clumped together to form a morula. And in day 5, like this is most important because this has formed a blastocyst. So this particular layer that you see on the outermost layer, that will form a placenta. Okay, and all these cells that you are seeing on here, like this is embryonic stem cells. Only this blastocyst is going to form an entire individual when get implanted into the uterus. Then this one blastocyst is getting differentiated to form a fetus. So this is called totipotent. Now in this blastocyst, there are stem cells, embryonic stem cells, which are present, which can also form almost any type of cell type in the body. So like this entire blastocyst is going to form an embryo, and then and then into a fetus, and then be developed. So by just one cell, it is we are getting an entire organism made. So in that process, we are learning about a totipotency, which can, from one cell, it can differentiate into a complete organism. Pluripotent stem cell, it can form almost all cell type in the body. And then induced pluripotent stem cell is something like scientists in the laboratory are playing with embryonic stem cells and getting them get converted into an induced pluripotent stem cells. And totipotency is this. Now, for you to make it easy, you can see this, like this is one cell. Can you all see the video?
Yes, sir.
Yes, sir.
Yeah. So you can just see one cell, how it differentiates into, how it starts differentiating. There was only one cell in the beginning, but then like you can see like how these cells are getting differentiated and how they are forming an entire cellular monolayer. So this is how it happens inside our body, so that like from one cell an entire organism is made. At the same time, not every cell differentiate into every type. They have been given a specific task to so that they differentiate into a specific cell types, and based on that you have a capacity to form an entire organism. At the same time, you have a capacity of a cell differentiating into only specific cell type, and then you have a capacity of cell differentiating into only one cell. So with this, we are getting into the diagrammatic representation of what we have studied so far. Like the fertilized egg and the totipotent stem cells, you know, like they are totipotent, they can form into an entire organism. And this pluripotent, this cell can almost form every cell type in the body. And then in case of multipotent, like I said, these are hematopoietic stem cells, so they give rise to blood cells. And these are neural specific tissue cells, they give rise to cells of nervous system. And mesenchymal stromal cells, they give rise to connective tissue, bone, cartilage, osteocyte, and everything. Now, with all this comes something called stem cell niche. So all this stem cell should be residing in some area. Like you need to have a house for the stem cells to first start, and then to self-renew, and then to self-differentiate. They need to have a space from where it all starts. So that is something called stem cell niche. So what is stem cell niche? So niche is a complex organization of a stromal, that is interact normal non-hematopoietic cells interacting via an adhesion molecule within the stem cells. So you can see over here, like this is the niche, and then the stem cell is getting interacting with an adhesion molecule within the stem cells. So this red dot is basically the adhesion molecule where the stem cells are getting attached in the niche. and this is the niche containing stem cell. And there are various kind of niche available inside the body for different tissues. Now what happens is, when a stem cell is dividing in the niche, it forms two cells. Okay, and these two cells, one cell will remain as a stem cell and another cell will become a transit cell, which then will go and differentiate into the type of cell it needs to get differentiated. So what happens is, the role of transit cell is to divide rapidly but a limited number of times, so amplifying the cell numbers and its progeny differentiate to form a functional end cell of that particular lineage. So what it means here is, this two cell, out of this two cell, one cell will remain as stem cell, the other cell will get differentiated into a transit cell, and then it will go and form a mature cell of a specific cell type. It can be either blood, or it can be either tissue, or it can be either skin. So in that way, it forms a cell which is of a particular lineage. Now, what is a lineage? So before we get into what is a lineage, we will check it over here. Like, this is a long-lived stem cell in a special niche, slowly cycles. One daughter is a new cell, the other is a transit cell which leaves the niche. So this is actually the stem cell. It is producing one daughter cell, and this daughter cell is a stem cell which remains inside the niche, and one transit cell, it gets away from the niche, and then over here it comes, and then it amplifies the cell number, going through a limited number of division. Why limited number of division and why not unlimited number of division is something that we will be learning in the further slides. We will be comparing it with why stem cells are not cancer cells, and what is cancer cell and what is cancer stem cells. All that will give us a entire know-how of what is limited number of cell division and why a limited number of cell division is needed. Now, when this transit cell amplifies and it gets multiplied, it forms a functional end cell which do not divide. So when it forms a functional cell of a particular lineage, from there it can't further divide, but it can only do the work of that particular cell of what it is. If it is getting differentiated at the end of the day as a neutrophil, then it will only do the work of a neutrophil. It can't further divide and give rise to monocyte or anything else, but it will only do the work of a neutrophil, which is the first cell to act against an antigen that is coming inside our body. So in this way, from a stem cell niche, two cells are produced, two daughter cells are produced. One cell will remain as a stem cell because it has to self-renew and differentiate again, and one cell will become a transit cell, and then it will form a limited number of cell division happens, and then it will form a one functional cell which does not divide but only do the work of what it is assigned for. Now coming back, our entire talk for today is on hematopoietic stem cells and hematopoiesis. But before we got here, we had to know what is stem cell, what is stem cell types, what is stemness, and what are the different types of stem cells, and what they do, and how do they do the work, and what is stem cell niche. So we have learned all that now. Now we will be specifically talking about hematopoietic stem cells. Like I said, in this slide, I said two daughter cells. So this is actually a microscopic image of two daughter cells getting divided and then almost forming a transit cell, and one being a daughter cell of stem cells as such. Now, hematopoiesis is a Greek meaning to form a new blood, and it refers to a formation of blood cellular components. So like in blood there are RBCs, WBCs, neutrophil, basophils, monocytes, granulocytes, agranulocytes, and then like you must have heard about platelets, plasma, B cell, T cell, antibodies, plasma cells, and all that. So all that forms the form that comes under blood cellular components. So all these blood cellular components are derived from a hematopoietic stem cell. So the hematopoietic stem cells are nothing but multipotent stem cells, multipotent progenitor cells that can differentiate into limited cell types. Okay. Now, in a healthy adult, approximately 10 to the power of 11 to 10 to the power of 12 new blood cells are produced daily. So that is actually the capacity of how our body can work wonders. Like we just casually feel like we are not taking care of our body and we are not taking care of ourselves, but body inside of us is doing a lot of work. Can you imagine, like 10 to the power of 11 to 10 to the power of new blood cells are being produced daily, and then they can decide, say they need, all these cells need a home, and from there they actually take the shelter, they take the nutrient, and then they can grow and then they can differentiate. So what is that home is about for hematopoietic stem cells? That home is nothing but a bone marrow. So all these cells, they reside in the bone marrow. Now we need to know what is bone marrow, or how do we get the cells out of bone marrow for a transplant, and what does this cells do inside the bone marrow? How are we getting to get out, get the cells out from bone marrow? All that we will be seeing in the further slides. Now they are very unique as they have the ability to mature into all different blood cell types and tissues. They are self-renewing cells. So why am I emphasizing the fact that stem cell means self-renewal and self-differentiation? What is pluripotency? What is totipotency? What is embryonic stem cells? Why I'm emphasizing this again and again is because unless and until you are very clear with these concepts, you will not be able to understand the science behind transplantation, science behind cellular therapy. Without understanding this, it will be very difficult for you to keep your concentration on what I'm going to talk for next 4 days. So in that way, you need to be emphasizing the fact that you go back today, do a little homework of what I said, bring back and come back tomorrow and give an example of what is, give an example of one example of each, like one example of totipotent stem cell, one example of pluripotent stem cell, one example of multipotent progenitor cell, and one example of embryonic stem cells. So I need this activity to be done by you guys today. So when you come back tomorrow, you know not just the definition, but you also know the exact meaning of this. At the same time, you also know the practicality of how this definition is being taking place inside the human body. So coming back to hematopoiesis and hematopoietic stem cell, like they are unique and they have an ability to mature into a different blood cell types, like I said, and they are also self-renewing cells. Now, this is a most important aspect. So whatever that we have learned so far with respect to stem cell, even the cancer cell can do the same. In that sense, it can also self-renew, and it's like it can also proliferate indefinitely. So first on the left, there is a table that talks about the similarities. Okay, so like stem cell, they also proliferate indefinitely, and then cancer cells, they also proliferate indefinitely, and they are also self-renewal by similar signals, and they also self-renew, and then they are heterogeneous with different phenotypes, and then they are also again heterogeneous with different phenotypes. And stem cells can migrate to one place to the other. In case of cancer cells, during the last stage of cancer, that is called metastasis. That is when the cells, cancer cells, will start migrating from one organ to the other. Why it is called the last stage of cancer patient life cycle is because from one cancer, if somebody has a lung cancer, over a period of time, even with chemotherapy, radiotherapy, and all these therapies, like if the stem cells are like if the cancer cells are not killed, they have a capacity to start migrating from lungs slowly into the intestine, small intestine, large intestine, liver, and they can actually keep migrating from one organ to the other, and that is called metastasis. So the only difference here between a stem cell and a cancer cell is stem cells, they migrate, cancer cell, they also migrate, but during the time of metastasis. And they also express telomerase. Telomerase is what? Telomerase is an enzyme which is basically making us grow old, and that because every birthday of years the telomerase is undergoing a cell cut, wherein like the ATGC sequence, the repeat sequence in the telomerase end is getting cut. That's a reason the length of the telomere is getting cut, and that is the reason why we age. So in case of stem cells and cancer cells, they have an enzyme called telomerase, which actually makes sure that there is no breakage of the repeated sequence in the end of the telomere. So the telomere is not getting cut, but with the help of telomerase, the cells can self-renew as well as self-differentiate. And this property is there in both stem cell as well as in cancer cell. And have an extended telomere repeats. Here also, in case of metastatic cells, when they are differentiated, when they are migrating from one organ to the other, they also have extended telomeric repeats. And this stem cell and cancer cell both can differentiate, both can be tissue specific and undergo organogenesis. And case of they also undergo apoptosis, which is nothing but a cell death. So how can you differentiate? Now we have learned what is stem cell, and we have compared all the features of stem cells with the cancer cell. Now, like, you will be a little confused how stem cell is different from a cancer cell, because all the properties are almost like matching with each other. In that way, we need to further get into the integrities of the differentiation, because we need to know that. So on the right, you can see the table B, the normal stem cell property versus cancer stem cells. So what is cancer stem cells? They are the cells which actually grow unlimitedly, and then they induce a cancer or a tumor. So that is nothing but a cancer stem cells. Now you need to know the difference between how a normal stem cell work and how a cancer stem cell work. And this differentiation will give you an idea of how different they are, how similar they look, but how different they are. So normal stem cells is extensive but limited self-renewal capacity. And in case of cancer cells, they do not have a self-renewal capacity. In that sense, they keep unlimitedly multiplying. But in case of normal stem cells, like we have seen, like in case of multipotent cells, they differentiate to a certain extent, and then at the end they form a mature cell. From there they can't divide. But in case of cancer cell, it is completely different. They keep unlimitedly self differentiating, and they have a self renewal capacity. And then, like, organogenic capacity, in case of normal stem cells, it has an organogenic capacity, which means what? It can form an organ. Like we talk about hematopoietic stem cells. So all these stem cells can form a hematopoietic stem cells, which can give rise to a bone marrow, and then like from bone marrow you have all these cells. But in case of cancer cells, they have a capacity to form a tumor. And it has, in case of normal stem cells, they have a highly regulated self-renewal and differentiation. But in case of cancer cells, they are completely opposite. There is no control over the self-renewal and differentiation, so it is highly irregulated. And in case of normal stem cells, they are very rare in normal adult tissues. If you, like, we are all adults in that way, like if I take your blood of peripheral blood from your body now, and then like I look for the stem cells, the stem cell count will be very less. Okay, why it is very less and how we can bring the stem cells into the peripheral blood, we will get into the further slides. But in case of cancer stem cells, they are infrequent or rare. Within the tumors, they are very infrequent. And they can be, the stem cells can be identified with markers, surface markers. Like when I say CD34 hematopoietic stem cells, or more specifically like CD4 or CD8, these are all markers which have a specific cell surface marker on the cell, through which we can identify, okay, this is a B cell which has an antibody, which produces antibody, so it is a B cell. So it has a specific cell surface molecule. In case of cancer cells, they are similar types of cell markers that ordinarily in the same tissue. But the problem here is, like, they are more or less same, but they are highly different in their nature of expression. And normal stem cell has a now normal karyotype. So what is karyotype? Hello?
The structure and presentation of chromosomes.
Okay. So what is a normal karyotype is all about? So you are like all of us have a normal karyotype. So what it is, like for female we say like it is 46XX. In case of male we say 46XY. But in case of cancer cells, they have a abnormal karyotype. It can be like, you must have learned about Down syndrome, Klinefelter syndrome, and then like Turner syndrome, wherein like the karyotype is abnormal. Like either they have an extra X or an extra Y, or they have problems in the abnormality in the chromosome in terms of inversion, deletion, and or like insertion and all that. So that is what is normal karyotype and abnormal karyotype is about. So in case of stem cells, they have a normal karyotype, which like based on the donor, like if the stem cells are derived from my body, they will have a karyotype of 46XY. And then if it is taken from a female, it will be having a karyotype of 46XX. So now, like in case of normal stem cells, they are questioned most of the times, and it means like they are very active, and then like they can differentiate and self-renew. But in case of cancer cells, they are less mitotically active other than other cancer cells. Like cancer cell, they can keep like they are mitotically active, but cancer stem cells, they are not as such. And in case of normal stem cells, they have a capacity to generate normal progeny with limited proliferative potential. That is what we learned earlier. Like multipotent progenitor cells give rise to limited number of cell types, wherein in case of here, it is completely different. So this difference is what you need to know before we learn more on stem cell therapy, stem cell transplant, and like all the cellular therapies, and then all the latest technology that is happening, all will have the same basis covered. So this is a very basic thing that you need to know for to proceed further on your quest of regenerative medicine knowledge that you are going to acquire in next 5 days. Now, so we know like what is stem cell, what are the types of stem cells, and how they differentiate, how they are different from cancer cells, and how they are different from cancer stem cells. And with this note, we also learned about what is hematopoiesis, that is like blood forming like phenomenon, wherein like it forms all the hematopoietic blood cell types in the body, which we have inside our body, and through which they actually give rise to all the blood stem cells, which actually take care of us during infection, during transplant, during cancer, and all the other infections. Now, we are going to be talking more specific on hematopoiesis. So, hematopoiesis is nothing but a blood forming phenomenon that we learned. Now, what is hematopoietic stem cells? How they form? It's very simple. It is like I spoke about lineage earlier. I said like lineage is like getting differentiated into a specific cell type. So there is something called the hematopoietic stem cells, progenitor cells. They actually, like hematopoietic stem cells, they divide to lymphoid lineage and myeloid lineage. So this lymphoid lineage will give rise to all lymphocytes, and myeloid lineage will give to rise to RBCs, thrombocytes, granulocytes, and everything. So what it is? This lineage is nothing but a progenitor cell dividing, and it is committed for a self renewal, and with a specific limited number of cell division, it will divide and then it will form a specific cell type, which then will not divide but will only do the work of what it is assigned for. That is progenitor cells. So one cell will remain in the kitty and the other cell will travel as a transit cell, and that is a cell which gets divided and then differentiate. So in lymphoid lineage, all these hematopoietic stem cells will divide into lymphocytes. And in case of myeloid lineage, it will divide into all other blood types. Now, it's the same diagram, but same concept, but in a diagrammatic representation, in which it can make you understand the hematopoiesis principle little more. Like you have something called hematopoietic stem cells. This is a cell, and then like you have a lymphoid progenitor cell. It is being progenitor cell, and it is giving rise to a specific cell type. And once it has given rise to a specific cell type, it will stop dividing, and then it will only function as what the cell has been assigned for. In this case, this is a T cell, and these are the markers associated with T cells: CD3, CD2, CD4. So if somebody is talking about CD3, they are talking about like I have enriched CD3 cells and I am looking for CD3 cells in flow cytometry. They are talking about T cells. And if they are talking about CD19 or CD20, then they are talking about B cells. Okay, and they also form natural killer cells. Now, this is a common lymphoid progenitor cell differentiating into a specific cell type, and this is what it is. It is all lymphocytes. Okay. Now, in case of myeloid, there is something called granulocyte macrophage progenitor cells, which give rise to monocytes, and then that monocyte gives rise to macrophages. And then this progenitor cell will differentiate into neutrophil, eosinophil, basophils. All these are granulocyte markers. So there is, you must have learned earlier, like what is like granulocytes and agranulocytes. You have granules, lot of granules inside the cell, and you don't have any granules. So in this case, like this monocyte is getting differentiated into macrophage, basophils, eosinophils, and neutrophils. And in case of erythroid progenitor cells, you know what is erythrocytes. So that is RBCs. So where these RBCs are coming from? They are coming from erythroid progenitor cells, which are giving rise to only erythrocytes. And in case of megakaryocytes, they are differentiating and giving rise to a mature platelets. So you know what is the function of platelets. So this is hematopoiesis, wherein this one hematopoietic stem cell is differentiating into lymphoid progenitor cell that gets differentiated into lymphocytes, and myeloid progenitor cell that gets differentiated into granulocytes like neutrophils, eosinophils, basophils, macrophages, and then monocytes, erythrocytes, RBCs, and platelets. So this is actually the way in which the cells are getting differentiated. Even though these cells have the capacity to self-renew and differentiate, because of the limited cell number of divisions that they have, they differentiate into a specific cell type, after which they do not divide. Okay, this is the concept. They have a capacity, but due to its limited capacity, they differentiate into a specific cell type and then they stop dividing. So this is the basis of hematopoiesis, in which all the cells are getting formed inside the bone marrow. Now, we will be specifically talking about first myeloid lineage, and then we will talk next about a lymphoid lineage. So myeloid is a tissue of bone marrow. They have differentiated from hematopoietic stem cell, then the myeloid progenitor cell. And they are this myeloid lineage, they are associated with innate immune response. Innate immune response is like it is there ever since you are born. It is like inborn natural immune system that is already there in place. And they differentiate in the bone marrow into RBCs, platelets, monocytes, macrophages, dendritic cells, all form a component of innate immune response. So innate immune response is basically through myeloid lineage, which is coming from hematopoietic stem cells. Now, lymphoid lineage. They are composed of T cells, B cells, and natural killer cells. And these cells are the ones that they actually kill the foreign cells and defend our body against cancer, pathogens, and foreign matter, which you might be knowing earlier. Now, and these cells, they circulate in the blood, lymph fluid, and found in body tissues like spleen, thymus, bone marrow, and lymph nodes. So all these are the sources of lymphoid lineages and lymph nodes, like lymphoid lineages cells, and these cells are basically for adaptive immune response. That is, once the foreign substance comes in, the body is aligned to that and it starts producing antibody, and then it fights that antigen, and during the fight it is producing a memory cell, so that it is keeping that memory cell in the kitty, and when the same antigen comes in, then this memory cells will act and they will start producing antibody. So this antibody will go and fight the antigen. So that is an adaptive immune response, wherein like we are getting adapted to the virus, and because of that adaptability, the immune system is getting primed and it is learning from the priming which is being done. And once it learns, it is also storing a memory, and that memory is helping us to fight the same antigen when it comes next time into our body. So all that is basically happening of cells which are derived from lymphoid lineage. Now, with all these talks, let's have a little activity done. So we are now like going to do some activity like making red blood cells. So you all can unmute, and you can how you can just give me an answer. Like if we like how to count, like if you are counting RBCs, like how long it will take for you to count RBCs and make RBCs as such in our body. So like if we count 20, if we count number 1 2 3 4 5, like if we count 20 in 2 seconds, then it will take 2 days for us to count 2 million. Okay, and if we do count 20 in 3 seconds, then it will take 3 days. And if it's 4, it is 5 days. And if it's 5, it is 6 days. And if it is 6, it is 7 days. Now, I want you to guess, to make 2 million red cells in our body, how much time it does take for our body to produce this 2 million red blood cells? Can anyone give an answer or even a try? Hello? Hello?
Yes,
Sir,
sir.
about a week.
About a week? Okay. So any more answers? I hope everybody is not sleeping and they are attentive and learning. I hope I'm right. And I'm guessing like everybody is glued to the presentation.
So less than a day.
Less than a day?
Yes, sir.
Okay. So like any more guesses, any more answers? The girls are sleeping, I feel. Hello? It takes just 1 second to make 2 million red blood cells in our body. So that is actually the capacity that our body has to make cells. And you must be wondering like how efficient our body system is to make these cells and to differentiate these cells into a lineages and then start multiplying them and then making these cells. So it takes just 1 second for the body to get making the cells. Now the second activity is like now you know like it takes 1 second to make 2 million cells of 2 million red blood cells. Now like if I take each RBCs from the body and then start putting in a linear way, that is in a linear fashion, if you lined up all the red blood cells from your body, how far would they stretch? Like you have a road which is an highway over here, and then like if you are just keeping RBCs getting lined up, how far they can stretch?
Sir, about the diameter of the earth, or maybe more than that.
Yeah, like it is like 4 times the Earth's equator, which is like almost like 20,000 kilometers. So in that stretch it can actually, you can like if you take the body blood and then separate the RBCs and keep lining one RBCs in the like keep one lining in the RBCs like nearly 4 times the Earth's equator. So that is how like magnificent as well as gigantic our body is. But everything is like everything is put in a system wherein like it's like so compact but so complex. So with this two activities we are going further into the adaptive immunity and the innate immunity that I was talking about. So in case of innate immunity, like I said, all these cells like neutrophils, basophils, and all that, they are all inborn and they are there from the present, and they are there from your birth. And that these cells are very rapid cells and they have a rapid response to the antigen which is coming in. Okay. And in case of adaptive immunity, it is a slow response. It takes for the body to know what it is, and then it starts producing the antibody, recognizing it, and then start producing the antibody against it, and then it makes the memory cells and all that. And there are cells which are equally taking part in both innate and adaptive immunity, which is like natural killer cells and like gamma delta T cells. Now, like you all must like we are getting going through this pandemic, and you must have learned a lot about or heard about lot about COVID and everything. And why COVID is like in case of immunity wise, like why COVID is like the antibodies are not recognized in our body in first 3 days, 4 days. Why it is taking a week time for the body to actually know that the virus has entered? Is there any reason which you feel like there is something that is happening in the body when a virus is entering, and that virus is doing something which is very crooked so that the body doesn't even know that there is something with foreign which has come in. Like, what do you think is happening? Like in case of COVID, since we are in COVID pandemic and everything, like what is happening in the way, like the body is unable to even do an innate immune response, which I just told you, it is a very rapid response. So when there is an antigen, it has to immediately act against it. But in case of COVID, like in case of viral infection, like it doesn't happen that way, because the virus, any virus, it evades the innate immunity. It evades the innate immunity. In that way, the body doesn't even know that the virus is there. And once the virus, and during this course of time of 10 days to 12 days, the virus will start multiplying in the body, and then like it start producing the antibodies. And only when on the 12th day or on the 14th day, when you go for an ELISA test, then you can see the antibody level popping up, and that is like different antibodies are there, we will be talking later. But that it takes the virus to evade the innate immune response. By evading the innate immune response, it is getting the cells multiplied. And viruses are like gypsy, they actually need a home for it to come, sit, take nutrient, and then start dividing. So that is happening. For it to come inside your home, like it has to evade the CCTV camera that you have put in your home. So that is nothing but an innate immunity. So that is evading that, and then it has come in, and then it starts multiplying. And then over a course of time, the virus multiplication will lead to recognition of antigen surface, and then like the T cell, B cell activity happens, and then the adaptive immunity kicks in, and then antibodies are produced, and that antibodies are present in your bloodstream, in the plasma. When you take the blood out in the plasma, you look for antibodies, and that they are present. So this is a difference between an innate and an adaptive immunity, which is a very fundamental thing, but a major thing to know when you are learning about any type of blood cancers and other clinical condition which requires transplantation. So in that way, like if we are going to prepare a patient who is, imagine like who is going through, who's getting a treatment for leukemia or a B cell lymphoma, like his immunity is always going to be lesser compared to a normal adult. Why it is less is because like the cells are not getting produced, the right cells are not getting produced, and because the immunity is less, there are viruses, there are bacterial infection, and fungal infection, the recurrent fungal infection, recurrent bacterial infections are happening, and the patient is immunosuppressed, immunosuppressive, in the immunocompromised. In that way, like all this is happening, and hence the patient loses out the battle with the disease. Similar way, that is what also happens in COVID as well. Why people are dying and everything is because like the cells which are actually getting produced or which are getting primed, they are all pro-inflammatory cells which can actually damage the tissue. So virus is making an attempt to induce all the pro-inflammatory cytokines which are not required to be activated as such. By getting all those things getting activated, the virus is making sure that it is evading the natural pathway of getting caught by the immunity, by the immune response, but rather also doing a damage to the same body by making sure that they are inducing the pro-inflammatory cytokines and all that. And that is the reason why you have something called cytokine storm, and that cytokine storm is eventually getting the body lose its battle. And you must have heard and you must have seen like people are negative of COVID, but then like they died of comorbidities as well as like multiple organ failure, or like they are negative but then they just come to the death. All this is because of manipulation in the immune system that is happening by the virus, and that immune response, which has to be regulated, immune response, it's getting dysregulated, and with all this, they are actually evading the pathway. And same happens in cancer cells as well. And when we are going to talk about acute T cell, like B cell lymphoma, multiple myeloma, acute AL lymphoma, like leukemia or CML, and all that, all has one concept, that is the patient, and the patient has an immune system, and all the immune system has something called innate immunity and adaptive immunity. So how strong your basic is in terms of immunology, in terms of adaptive immunity, myeloid lineage, lymphoid lineage, is what is going to make you learn the entire concept in a practical way, rather than just mugging up the fact that myeloid lineage means like macrophages, like it's that it will not work, because there are going to be integrities of each cell doing a different function in different cancers, and each cancer will have a different impact on these cells. But the concept of innate immunity and the concept of how the cell function will remain the same, and that has to be thoroughly and detailedly studied and learned first, and then to be studied. Okay, so with this we are moving further. Now I said, I was just giving an example of how it works in COVID and everything. Let's see how it works. Like a pathogen enters the tissue through a wound, and then it is directly attacked by macrophages. Macrophages means myeloid lineage. Myeloid lineage means innate immune response. So it is a rapid immune response. So it is directly getting attacked by macrophages, and then the antigen of the pathogen are displayed on the surface of the macrophage, and that is actually that signals are displayed, and that is activating the T cells, and T cells will bind to this, and T cells, and then what happens is, like, the B cell also binds to the antigen, and then it starts producing T helper cells, and then it starts producing the chemical that cause B cells to produce clone of plasma cells, and the B cells will start producing antibodies. The antibodies will interact with antigen, and then they nullify the antigen. The antigen is destroyed. At the same time, while doing this, on the point number G, the T cell releases chemical that cause B cells to produce clone of plasma cells, and that is, we will learn what is plasma cells. And by this process of B cells producing the antibody, and then the antibody actually attacking the antigen, in that process the memory cells are produced, so that, like, when the same antigen comes in, the body knows how to react. And then at the end, the each plasma cell secretes more than 200 antibodies per second in the blood, and the memory B cells and antibodies remain in the blood and responds to further invasions by the same pathogen. So what is plasma cell? By this definition here, it already says that each plasma cell secretes more than 2000 antibodies per second. So this antigen is all over the body, which is spread. And for this to get tackled, you need to have the plasma cells, and that plasma cells will carry the antibodies, and then they get distributed across the body, and that antibody then attacks the antigen, and then it nullifies the antigen, and then then it makes sure that antigen is gone. That is the reason, whenever you have having any viral fever, or like any infection, the first thing that they do is take a blood, do it, and separate the blood, look for the serum. The serum has like plasma cells. In that plasma you can have antibodies, so we look for the antibodies test through ELISA or through PCR. Like, we look for the blood, then we look for the antigen. So all this is happening, all this tests are being designed in a way that body, when it is reacting to the antigen, it has to have different cell types, and cell types working in tandem to make sure that these cells, they do the specific function, but at the same time also coordinate amongst each other to produce a good cellular response against the antigen. So in that way, this T cells will release the antibody. The T cells, through the T helper cells, will make sure that the B cells are releasing the antibodies. That antibodies are getting produced at same time. Like, you have a cell mediated immune response and an antibody mediated immune response. So both of them are happening in tandem to make sure that the antigen is destroyed. So this is more important and a very basic component of immunology that you need to know to learn further on all the latest technologies and learn everything about how the body works, not just during cancer, but during any other infection that is happening inside the body, including COVID that we are going through. Now, the like B cells and plasma cells. So what is plasma cells? White blood cells that secrete large volume of antibodies transported in the blood and the lymphatic system. And then this B cells, they differentiate into plasma cells that start producing the antibody, and then they bind to the antigen and start the destruction, like I said. So this is a diagrammatic representation wherein, like, you have an antigen, and then antigen specific B cell receptor is formed, and then this antigen presenting bacteria, and then, like, you have the antigens here, the receptor is just binding to it, and then, like, you have the class 2 MHC and process antigens are displayed, and then you have lymphokines, and then the T helper cells are activated, and then it actually gives rise to plasma cells, and this plasma cells will start carrying all the antibodies that are required to destroy the antigen. Now, so with this, you have the B cell mediated immune response, which is nothing but antibody mediated immune response. So before we proceed into how this immune response is happening, what is being done against cancer, what we need to do in cancer patient, how are we going to take care of this immune response and the immunity build up in cancer patients when they have been given a high frequent chemotherapies, how difficult it is for them to produce cells, how they are prone to multiple EC infections, like the most common virus amongst Indian population is like cytomegalovirus. Cytomegalovirus, like, if you take the blood of all people in the meeting today, including me and Professor Kannan, like, when our blood is taken, and then when it is looked for CMV, cytomegalovirus, IgG antibodies, everybody will be immune to it and everybody will be positive to it, because that is a very common virus that will not have any effect on normal adult individuals who are very healthy. But in terms of any patients where their immunity is compromised, the first and foremost virus that comes and causes the infection is cytomegalovirus, which is a very common virus. 90% of Indian population is known to have this, and they will be having IgG antibodies produced. IgG is memory cell antibody, so I'm talking about that. I'm not talking about the IgM, which is produced first during the infection and not during the latent infection. But the latent infection is generally being taken care by IgG, which we'll be talking in immunoglobulins. So why this is more important, why to study this immunoglobulins and B cell mediated immune response, is because, like, you have a patient who is immunocompromised but and undergoing a chemotherapy. So we are trying to eliminate all the cells which are defective. In that process, we are actually making sure that we are vacating all the cells in the bone marrow, which is deficient cells. In that process, we are making sure that the patient is highly prone to infections, and how to make sure that their immunity in terms of innate immune response and adaptive immune response is working is something that we need to look at. For us to look further into this subject, we need to know the basics of innate immunity and adaptive immunity, which we discussed. Now coming into the B cell immunity, which is antibody mediated immunity. All that antibodies are produced through immunoglobulins, and this immunoglobulins have 5 different immunoglobulins, and you have different structures of immunoglobulins as well. I'm not getting into the integrities of what is there in the antigen presenting side, what is there in the antibody side, whether it is lambda chain, gamma chain, kappa chain, all that I'm not getting over there right now. We can have a separate session for it maybe later, but right now we will be focusing only on immunoglobulins, which are specific in specific terms with the hematopoietic stem cell transplant and the clinical conditions that we are going to talk. So in that way, IgG will take care of bacterial and viral infections, and IgE is parasite and allergic infections have been taken care, and IgM is the first antibody that is produced by the body against any antigen which is coming in. That is the reason, like in case of COVID, like the first thing that they do is you have something called IgM and IgG. So IgG is the memory cell antibody. So you can, like when I tell that 90% of the population is IgG positive, it means that the body has got CMV virus and our body has tackled that virus, and we have a memory cells in IgG for specific for CMV. And when tested through ELISA kit, we are getting to know that we are IgG positive, means there is an antibody repository which is already been built up in the body specific for specific for CMV. Okay, and IgA is basically on the body surface, mucous membrane. Like you must have heard, like when somebody, like when you fall down and you have a scratch, like there is an Indian way of saying, like apply saliva on the wound. Why? Like it has antibodies. What antibodies there on that? It is basically IgA, which is present in all the mucous membrane, like starting from the saliva, and then like it on the nasopharynx, like a linear column, and then getting into the esophagus, and then the small intestine, and even on the intestine you have the mucous membrane of epithelial cells and endothelial cells, which have this IgA expressed. So very common example of IgA is very simple. Like in childhood, you must have got oral polio vaccine. Okay, in India, oral polio vaccine is given. What it means like oral polio vaccine. So vaccine is given in an oral form. So when you are taking it in an oral form, like because you like when it is taken in an oral form, from the time you take it till it reaches the intestine, you have on the mucous membrane the endothelial cells which will start multiplying against that vaccine which is there, and then it will start producing IgA antibodies, and that IgA antibodies it's there in entire mucous system, which is mucous membrane, starting from your mouth till the intestine. It is there. That is the reason why, and the probability of this cells getting multiplied in the intestine because of the mucous membrane, it's very, very high. That is the reason why polio vaccine is given as an oral polio drops and not as an intradermal injection in Western countries, where it is an inactivated polio vaccine which is given in an intradermal route. So this is an significance of IgA. And IgD is basically, like, not much is known about IgD, but it is being discussed and found that it also activates B cells. So now you know, like, what antibodies are getting produced for what and how they work, and what is the memory cells. Memory cells are nothing but IgG, and the first cell, the first antibody that gets produced in the body against any antigen is nothing but the IgM. Okay, so with this, we are getting into the hematopoietic stem cell transplant. So with all this 1.5 hour thing, we have learned what is stem cells, what are the types of stem cells, how do they do their work, and what happens inside the body as an innate immune response, adaptive immune response, how hematopoietic stem cell lineage is getting made, why they are specific lineages which are there, what is the function of lymphoid lineage, what is the function of myeloid lineage, all that we studied. For what? We studied, like we learned this to know the integrities of how they are getting used or what is their utilization and how they are going to work and how they are working in a patient who is undergoing a stem cell transplant. Now, before that, we get into that transplantation, there is something called a small activity. Like, again, I need everybody to answer this question. Like, somebody is sick, they get admitted in the hospital, and then the electrolytes are given as an intravenous route to them. So that is nothing but introduction of a solution into your body. Okay, that is nothing but infusion. Now you need to know this terminologies, because there is a huge lot of difference between infusion, transfusion, transplantation, and then in terms of transplantation, you have hematopoietic stem cell transplant, and you have something like organ transplant as well. So you need to know the difference between all these terminologies for you to further understand the subject in a crystal clear way. So infusion is nothing but you are administrating an electrolyte or an saline in an intravenous route through the vein into your body. Now that is infusion. Now transfusion. Transfusion is nothing but the blood. Like if you are O positive, you can donate the blood to anybody. So your blood is getting donated. That is through the vein into the next somebody else's body. That is transfusion. Now, if you see this picture on the right, this is also blood only. Okay, and but this is stem cells. This is all blood stem cells, but there is no difference between the blood stem cells and a blood. It is also a blood only. All this is looking like a blood, and even this package that you see is also same for any blood bank. Also, if you have gone to a blood bank, or if you have seen a blood bag of getting stored cryopreserved in minus 80 degrees Celsius, you will have the same kind of pictorial as well as representation, wherein like it will also look like blood, but there is a difference between a transfusion and a transplant. This is also blood, but when we give this and when we term this as a blood stem cells, but when we give this into a patient, we don't call it as a transfusion, we call it as a transplant. Why? Do you think there must be a reason?
Sir, I think the because in this case, in the bag you are saying there are stem cells, right? So
Mhm.
the stem cells have a purpose.
Mhm.
Whereas in case of transfusion, it's like when a person faces a blood loss, so for that
Yes.
recovery of that person, you are using transfusion
Yes, yes,
like that.
absolutely, absolutely correct. In case of a blood loss, like when a blood is given, it isn't like a, it's not a permanent fix, but it is a temporary fix, wherein like the cells are healthy and you're replacing them with healthy cells because of a loss. But in case of transplant, the cells inside the patient body is defective, and that is the reason why you want that to get replaced. And then once get replaced, you want them to self renew, start differentiating, and then start producing the cells like what it was earlier in the patient's body when he was normal. So that is the reason why you are changing the entire, it is like getting the flat, changing the entire interior decoration and making it custom for yourself. It is something like that. in that case, the stem cells will go in, they actually differentiate, they will just first adhere, and then they will have to start multiplying self, and then they'll have to self renew, and then they'll have to differentiate, and then they'll have to differentiate into specific blood stem cell types, which then have to do the work, and everything is getting replaced. So that is the reason why it is called transplantation. Now, in the transplantation, there is two different things: hematopoietic stem cell transplant and an organ transplant. So, in case of organ transplant, you might be knowing like kidney transplant and then like liver transplant and all that. In that case, over there, the like you all for all this transplant, there is something which is very unique, which is most important. What is that? Do you have any idea like what would be that one factor which is the most important factor for or during a transplant? Any
So
guesses?
HLA matching, I mean MHC.
Yes. Yeah. Yes. 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 HLA matching. So HLA matching is something which is very unique, and it has to be unique. And even in amongst it, like amongst the people in the meeting as of now, like we might have O blood group, 6 people, like B blood group, B positive, A positive, all that we might have at least 2-3 people amongst this group. But if we go into the integrities of our blood, and then when we go and check the HLA typing, you will have different HLA alleles amongst all our people, and the probability of amongst us finding a match is very less. So that is something. HLA is something which is a most important aspect. In case of organ transplant, it is not that effectively needed, but it is needed. But in case of hematopoietic stem cell transplant, since we are talking about cells that are going to take care of our entire immune system, entire body, then you need those cells to have a correct HLA type cells. Only then it will get transplanted and will start doing the work. What happens when HLA matching is not done? Imagine like your HLA is different and mine is different, but your blood stem cell, your blood is given to me for a during a transplant. What will happen? Like when some when a blood group like when a HLA mismatch product is given inside my body, it will immediately reject. So that is nothing but graft wise host disease, GVHD. So we will be talking more on that aspect as well. So a transplantation means the cells are also getting replaced. While the cells are getting replaced, the older cells are getting killed, and there is a conditioning. There is a period called conditioning where that happens in the hospital in a air conditioned, air controlled environment, which we will be talking in 2 days later. What is that air conditioned and air controlled environment, why it is needed, and everything. In that room, the conditioning happens. What is conditioning? It is nothing but reducing or removing all the deficient cells which are getting produced, that is basically causing the cancer. So during that time, like all the cells are removed, and then by giving the high end radiations, the cells are killed, and then your body is completely out of bone marrow, where the cells are not, even the deficient cells are not there. Now your body is all set to receive a cells from the recipient, or it can be a autologous or from your own body also, or depending on the clinical condition that we are going to talk. So this is the introduction about hematopoietic stem cell transplant. Now, so this, before we get into the subject as such, you need to know a little history of when did this happen, why did this happen, like how did this happen. And like this is like the first bone marrow transplant happened in the year 1956, and the cord blood transplant happened in 1988 in France, and peripheral blood stem cell transplant happened in 1986. So we will be talking about the all this transplant definition of it is in the next slides, but you need to know the history. That's the reason like why this slide is more important. The bone marrow first transplant with respect to cancer that happened in 1956, and this is a guy, Donald Thomas. He is the first guy who actually did a first successful hematopoietic stem cell transplant for the treatment of acute leukemia in 1956, and he received his Nobel Prize in 1990. So you must know, for any transplant work that is happening, he is a godfather of stem cell transplant, and his name is Donald Thomas. And this particular cartoon is basically the timeline of the stem cell transplantation. I'm not getting too much into this because not needed as of now for you guys. Now, what diseases can be treated by hematopoietic stem cell transplant? Now we are saying like hematopoietic stem cells means they can divide and differentiate into only blood cell types. So the disease that we are looking will also have to be having a malignancy or a defect in the blood. So that is the reason why we have all these diseases like aplastic anemia, thalassemia, sickle cell anemia, acute lymphoblastic leukemia, CML, chronic lymphocytic leukemia, non-Hodgkin's lymphoma, Hodgkin's diseases, autoimmune disorders like multiple myeloma, acute myelogenous leukemia, and then little selected solid tumors as well. Now, in case of children, it is like the probability is like acute leukemia is very prominent in children, and then MDS, lymphomas, bone marrow failure syndrome, hemoglobinopathies like thalassemia, sickle cell anemia, and then immunodeficiency and metabolic disorder and histiocytosis and others. In case of adult, like 59% is again acute leukemia, and then MDS, and then like this is lymphoma, and then plasma cell disorder, bone marrow failure, and other non-malignant disorders. Now, in case of children, it like we are like this we spoke about in general, like what are the diseases that can happen in children. Now, autologous and allogenic. In case of autologous, what is autologous, what is allogenic, we'll talk a little later, but these are the diseases that is basically been basically catered for in terms of hematopoietic stem cell transplant in children. Now, like types of stem cell transplant, you might have your own cells, that is autologous. You can take your own cells and then inject it back into yourself. Allogenic means getting it from a donor. That donor must be a different person. So he can be a sibling, and it can be an unrelated donor or a parent or a relative, or it can be even from the source called umbilical cord. Okay. And what are the sources that we are talking about? So in case of sources, it is bone marrow, peripheral blood stem cells, and umbilical cord. Okay, these are the three things. These are the three sources of hematopoietic stem cells that we can get, and these are the three which are being currently used. And there is something called haploidentical transplant, which I'll be talking exclusively on day 4, where I'll be talking on haploidentical transplant and the latest technologies associated with that. So right now we will focus on bone marrow, peripheral blood stem cells, and umbilical cord. Now, first is bone marrow. Like I said, like all the types of stem cells are getting made in the bone marrow. So that is a standard source. And then we select the marrow because extensive clinical data, because the first transplant happened in 1956, like so more than like 70 to 80 years of data of clinical data that we are having, and it is well known fact of getting bone marrow use. So we have that. And then extensive information is about the bone marrow experience as well. And peripheral blood stem cells, mostly in autologous transplant, rely almost exclusively on peripheral blood. So autologous, like I said, it is of bone stem cells. So in most of the cases, when we are looking for an autologous transplant, it is basically from peripheral blood stem cell transplant. Like earlier I told you, like when you take the blood of us right now, and when we look for stem cells in a peripheral blood, it will be very low. Now we are talking about peripheral blood stem cell being a source. How are we going to get the stem cells which are in the bone marrow to come into the peripheral blood? There is a video which is coming in the next slide, which will explain you what happens when you inject a colony stimulating factor, which then propels the bone marrow from the which then propels the stem cells from the bone marrow into the peripheral stream. So that is the reason why peripheral blood stem cells are the source. More rapid hematopoietic recovery, decreased cost because there is no need for you to look for a donor or for a cord blood, wherein like you have your own cells and then you can go for it. We also use this method for certain instances for allogenic transplant in pediatric cases as such. And this is now this like this is a stem cell like transplants. Like you can see the white blood cells and then getting differentiated into like the stem cells are getting differentiated into white blood cells as well as red blood cells. Yeah, this like sorry, yeah, the cells maybe weakened by the cancer treatment because like I said, like when you are having a deficient cells, like they will be going for a chemotherapy, so they will be weakened by cancer treatment. So that is the reason why they are deficient. So this is autologous transplant, basically where the stem cells are taken from your own body and then they are getting injected back. So this is basically after high intensity chemotherapy is given to kill the cancer, and over here like you can see that the blood is coming like it is getting replaced. Autologous transplant is happening, it is taken from your own body, so it is coming back. And then like in case of allogenic, like because it wipes out the bone marrow, we have to get healthy cells from the donor. So if you are unable to use your own cells, you will have to find a donor. So donor has to closely match, like you said, the HLA typing has to match. So this is actually a little cartoon on how the donor cells will come handy for a transplant. So you can have a sibling who might be a perfect match, but the probability of HLA match is also not guaranteed as such. So 25% of similarity is there, but in most cases, like the sibling can be a donor, but in most other cases, sibling can't be a donor. But there is also a very good thing that you can have somebody who is not related to you, but they can be an unrelated donor where your HLA and his HLA is matching. Because because of the HLA match, he then can be contacted and then he can give he can give peripheral blood stem cells as such. So they may be found. Like, how will I find this guy? Like there is something called registry, like a blood bank. There is something called registry where I will be going to a registry. Then the blood test will be taken for studying all my infectious disease screening. I need to be completely negative for all the infectious disease screening. Once that is done, then if I'm eligible for the blood to be given, then my HLA sample will be taken, and then like it will be typed for HLA typing, and then that data will be kept in the repository. And once there is a request, we will be running through the repository to find a match. And if there is a match between me and a recipient, I will be called, and then I'll be given an injection to get the stem cells come out of my bone marrow to the peripheral stream. Then I'll be going an apheresis method where the stem cells will be harvested, and then like it will be transported to the recipient for the transplant. Now, the third source is umbilical cord blood. So, umbilical cord blood is a good choice because in most of the cases, like once during the birth, like of the child, the umbilical cord is discarded, and it is a rich source of stem cells, not just a rich source of hematopoietic stem cells, but also adult stem cells as such. So the HLA mismatch, like I said, it's also better tolerated, because in case of bone marrow, you need to have an HLA match of 6/6 between the donor and the recipient. And in case of cord blood, it can be only 3/6 as such, and it is available more quickly than a bone marrow or a PPSC and related donors, and also reduces graft-wise host diseases. Now, this was actually the first transplant that we did in Tata Main Hospital in Jamshedpur, and this is basically, this patient is of a B-cell lymphoma, and this was the first time the transplant happened in that hospital. We were so anxious because, like, once you can see on this, you can see the blood bag, a bit of blood bag, and then the blood bag is not actually visible, stem cell bag is not, but the syringe over here is like, it also has a bit of stem cells here running over it. And then like we are monitoring the O2 level, SpO2 level, and the heartbeat, so that like the patient is stable or not. Patient was given grade 1 anesthesia, and then this entire setup where it is happening in a clean room environment, which is air controlled and highly controlled in terms of air infection, because as I said, the patient is already immunocompromised, so the probability of patient getting a disease is very high. So he or she can't be put in an environment where the possibility of getting an infection from the room is high. In that way, the entire facility is basically a clean room environment. What is clean room and everything we'll be discussing on day 5. So this is it, and this is every transplant physician's wish list. Like in case of bone marrow, the bone marrow is taken from the crest, and then in case of peripheral blood, the donor is actually donating the thing through apheresis. This is an apheresis machine through which the peripheral blood stem cells are being harvested, and then we are running through the CD34 hematopoietic stem cells. We enumerate it, we find it, and then it is given to the patient. Like you can see, it is also similar to blood transplant only. You can see the blood over here, which are all blood stem cells. They are all stem cells, but they look more like a blood only. And then like the patient has received it, he is stable, he is happy. So this is actually every transplant physician's wish list. Now, like I said, the CD34 progenitor cells or hematopoietic stem cells, they are formed in the bone marrow, and then like they can be either taken from the bone marrow. In most cases, it has to be taken from the peripheral blood. So how to make sure that the CD34 cells carry from bone marrow, how they are carried to peripheral blood stem cells, peripheral blood. So that is something like this is a video. Like you have the stem cells, and like I said, they are present in the bone marrow, and they reside in the bone marrow, and this is where they are getting resided. And here you can see the peripheral blood stream running over the thing. And in the bone marrow, you have two specific cells called stromal cells and osteoblasts. So on the blue side is osteoblast, and this star-shaped cell is your stromal cell. So these cells, they actually attract stem cells. How do they attract stem cells? Is basically through a factor called SDF1. Through that factor, they make sure that the stem cells are aligned and collected, and they are present in the bone marrow. And these cells then are there in the bone marrow because this SDF protein will form an association with the bone marrow endothelial cells, which are actually making sure that those cells are staying in the bone marrow by making sure that this endothelial cells will have a receptor called CXCR4. So this SD1 will come and bind with this CXCR4, and with this binding, it is making sure that all the stem cells are present only in the bone marrow, and they self-renew and self-replicate and self-differentiate, but their home is only bone marrow. Now, for us to get these cells out from the bone marrow and to make them flow in the peripheral blood, there is something called granulocyte colony stimulating factor, which is injected into the donor or a patient, which then will go into the body stream, and then it will actually produce three enzymes like neutrophil elastase, and then casprin G, and then MMP9. So these enzymes will then go and cleave that SDF and CXCR4 bond. So once that bond is broken, these cells are breaking that bond between SDF1 and CXCR4, and thereby making sure that the stem cells from the bone marrow, they are getting into the peripheral stream. From that peripheral stream, they are actually visible. They are then easy for us to collect by doing an apheresis procedure. So by giving us something called granulocyte colony stimulating factor, the colony stimulating factor is injected as an intradermal or intradermal injection over a course of 5 days. Like if I'm going to be the donor, if I have to give the cells first, I'm an adult, so in the stem cells in the peripheral blood will be very less. So I'll be given this injection over a course of 5 days. Every day I will be given this injection exactly after 24 hours. And on the 5th day, I'll be admitted in the hospital. Then I'll be going through an apheresis procedure, because during this course of 5 days, the cells from the bone marrow are already there in the peripheral stream of me. From there, I'll be taken for apheresis method. Then through apheresis, stem cells are separated, and the other cells, RBCs and everything, they are given back again into the body. Now, before we talk about the hematopoietic stem cell, progenitor cell, transplantation and everything, you need to know how does a CD34 hematopoietic stem cells look, because this is a very latest video wherein this is the first time a CD34 hematopoietic stem cell without any fluorescent labeling through a technology called nanolive, along with an association of Lonza, where this human cord blood hematopoietic stem cells were procured from Lonza, and then this company called nanolive, they, through their technology without even fluorescent labeling, they found the hematopoietic stem cells, which are CD34 hematopoietic stem cells, which are very, very rare to find as such. So this will give you an thing of how CD34 cells really look like. So all this circle of cells are basically CD34 hematopoietic stem cells, and they are migrating. So they are migrating towards the tissue of concern, wherein from there they can go divide, then regenerate, and then give give birth to certain types of stem cells. So this is actually a very good representation of stem CD34 stem cells in the body, and you can see how these cells are migrating. So now going back, why CD34 cells are important. So CD34 cells are antigens expressed on the surface of all hematopoietic stem cells. And enumeration of this will yield how stem cells have actually mobilized from bone marrow to peripheral blood. So number of CD34 cells will be the vital key when we talk about all the transplant of hematopoietic stem cell transplant. If a patient has to recover, and then if the patient has to make sure that the defective cells are replaced by good cells, then you will have to have the CD34 stem cells in good quantity and good viability, then getting injected into the donor for it, the stem cells to grow and then self-replenish. So accurate CD34 can help optimize the timing of apheresis collection. And then like it's a rare event analysis requiring specific gating instruction for detection. So unlike any other flow cytometry process of detecting cells, CD34 gating analysis of analyzing is very specific, and it is a very unique method of counting the stem cells. And it provides a standardized single platform flow cytometry method for enumerating CD34 cells. This method can be used to quantitate CD34 in samples of peripheral blood, apheresis products, bone marrow, and cord blood. The number of CD34 cells per microliter in the sample can be calculated by comparing the absolute target, which I'll be discussing later. Now, like I hope now it's 5 p.m. exactly. So tomorrow we will be basically talking about why CD34 is important again, and how it has been a brief animation on flow cytometer, and then like how the sample is prepared for enumerating the stem cells, and then like how the stem cells are enumerated. What this is actually the enumeration plot which says BD stem cell enumeration kit, and this is actually the entire diagrammatic representation of each plot. We will be discussing this. In each plot, we will be discussing the importance, relevance, and then like we will be discussing this of each plot, what is the relevance, and what is the scenario of India and US right now, and what is the scenario and everything. So, and then we will be also talking about the company where I head the lab operations. So what we do and how we are doing all this. And as of today, like anyone who keeps learning stays young. So with this message, I end the presentation for today. I hope it was informative. You learned something, and you also it was also interesting for you and gave you a chance to go back and read further on this subject. Thank you.
Thank you, sir.
Thank you,
Thank you,
Surya.
sir.
Yes.
Yeah. Hello. Just 1 minute, kids. Are you there? Yeah. Hello.
Yes, sir.
Don't worry. Lot of things might have gone above your head. Doesn't matter. You now actually heard a lot of new words today, new things, and this is one area where, as a biotechnologist, the lot of contributions have happened. He was talking about granulocyte colony stimulating factor. It is one of the big blockbuster drug, protein molecule made, and the company called Amgen actually made this product. They also made erythropoietin. So I want you to know that these are all some of the things. Why biotechnology became popular is because some of them. Don't worry, you got an overall feel. Don't worry, please write down whatever you haven't understood or you want to know much more about it. And whenever I spend time, I'll take you through the basics, like what is totipotency, what is pluripotency, what is, multipotency, unipotency, Yamanaka factor, because I'll take you through slowly. But what Surya has given today is a bigger picture, that there's a huge market for this. He showed you that, tomorrow when he discusses, he'll tell you how, because these are the techniques you have to learn if you want an employment. So basically what we are trying to give you is a bird's view of what is the opportunity available. Don't get scared by it, because Surya has learned it over the years. You are still the beginning, but once you know such an opportunity area there, you will slowly build learning it. So in the next year or two, you will become more confident. Then you will say, you'll take a informed decision whether I would like to be get into this, because many doctors will be looking for somebody who can purify these stem cells and give it to them for transplantation. So for example, AIMS has a group which only does purification of stem cells and give it to the doctor for the treatment. So what I'm saying is that this is another opportunity area. Don't feel very depressed that it went above head. Don't worry about it. At least you know that such a treatment is possible for, he talked about Donald Thomas, who was really the pioneer in the whole field, and he went through a lot of ups and downs in the 50s. And, we will learn about it. So what I want you to today appreciate is that please have a look at it. I'm sure you noted down. And over the next 3-4 days, many things will become clearer. I know some people were getting restless and probably left the meeting. But the idea is that we just gave you a flavor today. Now we'll elaborate on the flavor tomorrow. Surya will do it. If I'm okay, I'll be there at 10 o'clock tomorrow. Otherwise, Surya will talk to you at 3 to 5. Okay? Is that clear?
Yes, sir.
Yes, sir.
Okay. So but write down all your questions and send me. Okay.
I
Yes.
hope I did not scare them, because I started from the basics and then
No,
like.
I know. But the point is, Surya, they have never been taught like this.
Okay.
See,
Yeah.
they belong to a system which
Yes, we will.
where it is
And
exam oriented.
yes, sir.
So the
Yeah.
teacher only teaches what is
Yes.
given in the syllabus,
Yeah.
nothing more, nothing less, kind of thing.
Yes, but
But
yes, sir. That is something that we'll have to change
yeah, we
because
will change. So
yeah,
now there's no exam here. They have
yeah,
all the time to learn. Enjoy the process of learning how processes are done. Then you become wiser in the whole process. Is that
yes,
okay? Thank you, Surya, for a big elaborative. And tomorrow you're going to talk about how we enumerate CD34 on fluorescent activated cell sorter, which
yes, yeah.
he said is now that's a very important technique which is not taught to you by your faculty there. So please attend that tomorrow. You will know what is x-axis, y-axis, where the cells appear, CD34 on that plot. All that are very interesting because there are millions of cells and these cells are very few. So how do I know that they are there? So you will come to know tomorrow and then we will see it. Okay. So sorry for the heavy dose the whole day from morning till today, but that's how it is the best time to at least note down. You can learn at your own pace. Okay. Okay
Yeah,
then. Bye
like
bye.
if you have any questions, like we are open. Like I am, as well as Dr. Professor Kannan would be,
More
like
than happily.
more than like we are more than happy, and he would be much better in explaining the concepts than me. And you can ask me any questions during the course of the lecture, as well as like after the lecture as well. Like
Or
you
you
can
can
come
even
back,
you can put it
email.
in
Yeah,
the chat box also. You can put up your questions.
yeah,
Okay. So,
yes.
but don't just get a feel of it. Initially, it looked Greek and Latin. Then slowly you start understanding. All of us have gone through it. You think Surya understood it next day? No. He also took a lot of time.
Yes, yeah.
So did I. So all of us go through it. So don't get carried away that we are speaking so easily because we also learned it the hard way and you will also learn and tomorrow you will also talk like us. Okay.
Yeah, our intention is to only make this things very simpler to you guys, so that like you don't go through the same hardship of how we learned.
Exactly.
That's the
Exactly.
that's the
Exactly.
entire funda of how we are looking at this.
Yes, I totally agree. So I'll see you tomorrow.
Okay, guys, have a good evening, and I'm sorry if the dose was very high.